So there are major parameters, that factors that influence the chemical degradation of the pharmaceutical products. If you look into that, one is a solvent and it's a dielectric constant, and another part is a ionic strength of a solute, and acid-base catalysis, and general acid-base catalysis, and specific acid-base catalysis. So the last two we have already handled. The first one is also a factor we need to write. All the five you need to write when the chemical factors influencing the rate of reaction in the pharmaceutical degradation. So first we will be looking at the effect of solvent and the dielectric constant of the solvent. Most of the times our degradation will be progressing with the different rate of reactions orders, and the different rate constants they will be proceeding, and you will be having different half-lives and self-lives. We know that if the self-life is very less, we are going to have very less period of the time which will be present in the pharmacy store, and we cannot sell the product longer period of the time, so profits will also be getting reduced. We need to consider different factors so that by handling them, we will be able to come out of a solution to enhance the stability. So the reactions involving the ions of opposite charges. I repeat, if we have product A and product B, that is reactant A and reactant B, they have got the opposite charges. Generally, in that case, it is accelerated by the solvents with low dielectric constant. So the ability of the substance to store the electrical energy in the electrical field, depending upon that, it will be having. So reactions involving the ion of opposite charge are accelerated by the solvents with low dielectric constant. Example, rate of hydrolysis of the sulfate ester is greater in low dielectric constant, like methylene chloride, than in the water. So this one, it will give the idea that, so how the reactions involving the opposite charges are accelerated by the solvents with low dielectric constant. The reactions between the similar charged ions is favored by the high dielectric solvents. Reaction between the similarly charged means both are having a similar type of the charge. Such reactions are progressing, that will be favored by the high dielectric constant of the solvents. So reaction between the neutral molecule which produce the high polar transition state is favored by the high dielectric constant solvents. Most of the times a neutral molecule, they will be tend to exist in form of non-ionized state. But when you are putting this one in the high polar solvents, and then what will happen? You are going to have a ionization and making a ion formation that will make the progress of reaction. So this is the one which having an impact. Because of that reason, we generally prefer to use water, which is having a high dielectric constant, but sometimes it causes the damage to our product. So we need to replace the water with any another solvent. Most of the times it can be oil. Oil may be a sunflower oil, linseed oil, or any some oil, groundnut oils. The oily solutions, they will be having a better stability enhancement. We will be trying there. And if it is having a problem of hydrolysis also, then we will make a oily suspensions. And the last methodology we can use is avoid the solvent completely. What we need to do then, we need to dispense our product in the form of dry powder and ask the patient to reconstitute before use, and giving the expiry date second one and first expiry date before opening the bottle, and the first expiry date will be getting void when you are opening the bottle and reconstituting it. From that period onwards, date onwards, you will be having a second expiry date. It means before opening the bottle, how much is expiry date? After reconstitution, what is the expiry date of the product you are going to give, to expiration datings. Now you may get the question, sir, we have made the shelf life T90 and we have predicted the shelf life of that particular product. Based on that particular one, I am giving the expiry date stamped onto the package and I'm launching my product. In the case of 2, how can I proceed? This is actually the question which did not handle during our first order, second order and zero order kinetics, where we are calculating the shelf life. Generally what will happen, whatever the product you are selling in the powder form and kept in the bottle of HDPE, LDPE, that is high density polyethylene or low density polyethylene or glass bottle or any sachet, you are going to have a study of stability in that particular format only, and under that conditions of the storage, you calculate the shelf life depending upon the rate constant, how much we have got. So we know the initial concentration and we know the rate constant. By using them, you can calculate the shelf life. That already you know it. So 0.1 C0 by K, you are calculating it, 0.105 by K. So you know the formula, so you can calculate and you can put the expiry date. But the problem in this particular case is when the powder is getting reconstituted without using the solvent. Because when you are launching your product, you are not using any solvent. Because of that reason, it is highly stable, you are giving an expiry date. But when you are using a solvent, what is its effect? Obviously, it is having a higher effect. That is the reason you are not adding in directly into the bottle and you are selling it as a dry powder and asking the patient to do this step of addition of water. In that case, what will happen? You take the same product, make up with water before usage, shake it well, and we are asking in a similar format, we make the product in our lab and make up the bottle with water and make the suspension prepared or solutions prepared. Then we will start doing the shelf life estimated, whether it is in accelerated or in the real time. We keep the bottle there and at accelerated conditions, we conduct the testing and predict at room temperature, and we will get another expiration period that will be generally in the months or weeks. So this we are already putting in the label, rounding off to weeks or the months, not like years what we have posted previously. We are rounding up in the product, rounded up to years. Now we are going to have rounding up to months. And we will ask the patient after opening of this particular bottle, reconstitution, use before this date. That's what we need to give, because the solvent has got its own effect to degrade very faster, and it is producing the damage to the product. They cannot use after particular period of the time, because more than 10% of the drug will be degrading in the presence of the solvent in short span of time. Effect of ionic strength. Most of the times we will be using ions, either they are present in the form of drug or maybe they are having a excipients, they are present in the form of ions. The ionic strength, it will affect the progression of reaction. We need to consider this also. Accordingly, we can sometimes change our excipients to make its effect to the negligible. So ionic strength may affect inter-ionic attraction. Most of the times you will be having a two ionic species in the product, and they will be having an attraction if they are oppositely charged, repulsion if they are similarly charged. Okay, so in that particular case, increasing in the ionic strength, expected to decrease the rate of reaction between oppositely charged ions. So I repeat, increasing the ionic strength expected to decrease the rate of reaction between the opposite charged ions and increase the rate of reaction between the similar charged ions as per the Debye-Heckel theory. Debye-Heckel theory, it states that increasing the ionic concentration expected to decrease the rate of reaction between the oppositely charged ions and increasing the rate of reaction between the similarly charged ions. So this theory is based on three assumptions. Most of the times there are three assumptions. How the ions they act in the solution. Generally, we know that electrolytes are completely dissociated into the ions in the solution. Electrolytes are the charged species. When you are putting into the water and to make a solution, they will be present in the dissociated states. We know that there are two types of electrolytes, weak electrolyte and strong electrolyte. And there is an aberration between these two also depending upon the external conditions. So they will be present obviously in the water in the dissociated state. Second rule and theory, second assumption of this particular theory is solutions of the electrolytes are, if the solutions of electrolytes are very dilute in the order of 0.01 and m, so they will be behaving this particular one if they get ease, whereas the highest concentration solutions, they will be having a dissociation differences and deviations. But when they are dilute, they will be having highest dissociation. Each ion surrounded by the most of the ions of the opposite charges as an average. Because whenever we have got an ion which are present either a drug or a excipient, around it they will be attracting oppositely charged species, they will be present. The same phenomena we have already seen in the zeta potential, where nernst potential is surrounded by the diffusion layer, and the edge of the interface of the diffusion layer with the bulk, we are having a charge that is called as a zeta potential. So this is actually what is making there. Similarly, here also it is having an impact on the effect of ionic strength onto the different interionic attractions. So if we look into that, then we will be having further assumptions. Applying these assumptions, we can postulate some another thing, extension of it. What is that is, the Huckel and Debye-Huckel, they noticed that solution that contain the ionic solutes do not behave ideally even at the very low concentrations. We have already seen in the second rule, in the concentration to the dilution of 0.01, we have got a behavior of ionization. But generally they noticed that solution that contain ions do not behave ideally even at the lower concentrations. Why they are not behaving? Ideally they need to present in form of ions, but they are not behaving as ions even at the lower concentrations. What is happening there? In reality, they are not present in the ionic forms, and their activity is not same as that of the ions what they are expected to be. So while the concentration of the solute is fundamental to the calculation of the dynamics of the solution, we have already seen in the first is ionic strength, increasing the ionic strength expected to decrease the rate of reaction between the opposite charged ions, because they have got the greater tendency to react with each other, and they will be not available. That is, the drug moiety is not available for the reaction. So rate of degradation will be there. So this, but you need to think that the progression of reaction is involving a reaction between the two opposite charged, then it will progress. Then we will be having a concept called as a second order reaction, where we are having a presence of one moiety, it will be having effect. But it is considered in the drug excipient studies also. We are going to add this excipient and to the drug. If such any reaction with the charged species, it is resulting into degradation, we avoid that and replace with another excipient in the drug excipient studies. But generally, ionic strength of the solvent, which is expected to decrease the rate of reaction between the opposite charged ions and increase the rate of reaction between the similar charged, as per the Debye-Huckel theory. And Debye-Huckel theory, according to it, he has made the assumptions and postulated that even at the lower concentration, at the order of 0.01 normal, they are not behaving ideally, they are behaving in the real sense something else. So they theorized that the extra factor that termed is gamma. So that is necessary to calculation of the activity coefficients of the solutions. It is not the simple concentrations which are present in the solution. its activity coefficient matters a lot to progress of a reaction which is involving in a degradation. So this is what it has been postulated by them. And generally, if we calculate without considering these activity coefficients, we will be coming a erratic result when compared to your real result what you are expecting in the laboratory. So there is a deviation because these solvent moieties having opposite charges around them and getting attracted and making a differences in the reactivity of the particular ions, because the all ions, if we are having it, they will be present. If you see, they will be surrounded by the ions of the opposite charge as an average. So when they are surrounding it, they will be changing its ionic strength, and accordingly, activity coefficient will also be getting altered. Because of that reason, the progress of reaction is getting changed. So the activity is only proportional to the concentration and is altered by the factor is called as a activity coefficient gamma. This factor takes into the account of interaction energy of ions in the solution. It means even though they are present in the high ionic strength, and it is not going to involve if their activity coefficients are very low and low, because this coefficient will decreases the total reactivity of the ion species. So the interaction energy of the ions in the solution is actually depending upon this factor gamma. So this is actually helping a real sense, and we are making a predictions very easy. So this is what they have postulated, and we have already seen the general acid-base catalysis and specific acid-base catalysis in the presence of H plus and ions, and we have already seen that observed one which is depending upon the H plus and OS minus in the